let's go ahead and get started with chapter 30, which is on animal origins and the evolution of body plans. So we're talking kingdom Animalia here, which is huge, about 1.3 million extant species with incredible diversity in both morphological variation and in size. Now, there's so much variation that it actually sometimes is hard to find unifying characteristics, but there are some. One thing we can say about all animals is that they're multicellular. Now, this trait is not unique to the animals. There are lots of other organisms that are multicellular, like plants, like fungi. But all animals are multicellular. They also have a common nutritional mode, which is that they are heterotrophs that digest their food internally within a gut of some sort. Now, this is different than the fungi that would secrete enzymes and digest their food externally. Okay, so there are two cell types that we are only going to find in the animals, and those two cell types are nerve cells. Get my pointer here. So we have our nerve cells, and we have our muscle cells. My computer's not cooperating with me. Hold on. And we have our muscle cells. The ability to move and conduct nerve impulses are what give animals many of their unique characteristics, and it's one of the primary things that's going to differentiate them from plants or fungi. Now, there are some species that do not move during some parts of their life cycle, and there are others that lack a nervous system or have a very rudimentary nervous system that we call a nerve net. But these features of nerve cells and muscle cells are unique to animals. So what do all the animals have in common? Well, how do we put them together as a group? So let's look at this common ancestor here. So, well, one of the things that allows us to unite animals as a group are gene sequences, right? So we can look at the gene sequences and know, yes, these have an evolutionary common ancestor. But in terms of actual traits we can observe, we have extracellular matrix molecules, such as proteoglycan and collagen. Okay, so animals do not have cell walls. So the thing that gives an animal cell support and strength is this extracellular matrix around it. And so animals have these common molecules of proteoglycan and collagen, which hopefully you remember from 1406 as being part of the extracellular matrix. We also see some very specific cell junctions in the animals. So the tight junctions, the desmosomes, also known as anchoring junctions, and the gap junctions. So hopefully you'll also remember these from 1406. The tight junctions help make watertight seals between compartments. The desmosomes are structurally strong and help keep the integrity of tissues, and the gap junctions are large enough to allow ions or other metabolites to pass through for communication of cells. So most estimates say animals diverged from their fungal ancestors at least 675 million years ago, but some estimates put it more at like 1 billion years ago. Whichever date you want to talk about, our closest common ancestor is hypothesized to look like this. So this is a protist known as a cyanoflagellate. It is a colonial protist, and I know that this might look nothing like an animal, but actually it does have some striking similarities with one of the more basal groups of animals known as the sponges. And when we talk about protostomes, actually no, I take it back. We're going to talk about sponges in this chapter, so we'll talk about them a little later in this chapter. Okay, so here's the most commonly accepted animal phylogeny. And we will, in this class, through these chapters, be going through pretty much every group you see here. But first, I want to talk a little bit about animal development in general. So, there are some developmental patterns we see in stages that are common to all animals. So after fertilization, the single-celled zygote will undergo cleavage, which is mitotic divisions, that are not accompanied by growth in size. So this, it's not enlarging, but it is undergoing cell division. It's going to form a hollow ball of cells that we call the blastula. The center, that hollow center, is known as the blastocele. Okay, so the blastula is right here, and then here it is in cross section. So there's that blastocele. This blastula will undergo a process called gastrulation, where one end folds inward. And what this does is it creates layers. And these layers will eventually become the various parts of the animal, the organs, the appendages. So we're like a layered tube, kind of. And at this point, we refer to this stage of development as the gastrula. So this is the gastrula. And there it is. And you can see now they've colored, it's got two layers now, and they've colored the outer layer of cells blue and the inner layer of cells yellow. So as we talk about the different groups of animals, one of the features that's going to differ between clades is whether you have radial cleavage or what we call spiral cleavage. So radial cleavage is the pattern of embryonic development in which the planes of cell division are parallel and perpendicular. And in spiral cleavage, the planes of division are at angles, sort of oblique angles. So I know this is a little hard to envision. So here's an image of radial cleavage, and here's an image of spiral cleavage. So hopefully you can see how that is. So see how we're getting the cell division kind of at an angle here. I'm going to try to dig up a video we can watch in lab, because I think it's a little easier to appreciate this when looking at a video. So this process of gastrulation is really important because it produces tissue layers. So the initial infolding is going to create two layers of tissue, what we call the endoderm, which is the innermost. We call it a germ layer, the innermost germ layer. It's going to give rise to the digestive and respiratory tracts and structures that are associated with those features. And we also have the ectoderm. That's the other layer. The ectoderm, of course, is the outermost germ layer. And it's going to give rise to the skin, to the sense organs, and to the nervous system. So in some animals, but not all, there is a third tissue layer present. And this is another way that we'll actually divide up the animals when we're trying to make clades. So in some animals, there is a third tissue layer that forms, and this third tissue layer is called the mesoderm. So this is a middle germ layer. Animals that have it are described as triploblastic, meaning that there are 3 germ layers. This is going to give rise to the skeleton, circulatory system, muscles, excretory system, and most of the reproductive system. So here's a little compare whether we have 2 germ layers or 3. So monoblastic animals have 1 germ layer. And here we have a comparison of diploblastic and triploblastic. So a diploblastic organism has 2 germ layers, the endoderm and the ectoderm. This is actually the ancestral condition, and organisms possessing it don't necessarily constitute a monophyletic clade. A triploblastic animal has 3 germ layers, endoderm, ectoderm, and mesoderm. This is a derived synapomorphy, and it does unite all who possess it as a monophyletic clade. And we'll get into all that when we actually talk about the different groups. So within triploblastic animals, there are 2 distinct developmental patterns that become very important when we want to talk about different types of animals, animal groups. So during gastrulation, the hole that forms when the layers infold is called the blastopore. Okay. So when this infolding forms, we end up with this area here, which will be the lumen of the digestive tract. So the blastopore is either going to form the mouth or the anus, right? So you can think of this as being a tube, and this hole here eventually either becomes a mouth or it becomes an anus. In what we call the protostomes, the blastopore forms what will eventually become the mouth. That word protostome means first mouth. So the first hole that is formed becomes the mouth. In what we call the deuterostomes, or the deuterostomes, the blastopore forms the anus of the digestive tract. And later another opening will be created that will form the mouth. So each clade is monophyletic, so the protostomes versus the deuterostomes are 2 big important clades. Guess which one we're in? We're deuterostomes. So at one point in your life, you were just a ball of cells with a butthole. So gotta just kind of deal with that, I guess. That's the beauty of being an animal. Okay, we're going to break here, and we're going to pick back up talking a little bit about animal body plans. So see you soon.